Molt bon dia a tothom. Molt bon dia i molt benvinguts a aquest workshop d'intel·ligència artificial en biomedicina. Després ara farem la introducció en català, però després ja ens passarem a l'hora de les presentacions, ens passarem a l'anglès. Això, avui estem aquí amb aquest workshop organitzat entre la Facultat de Ciències, Tecnologia i Enginyeries i l'Institut IRIS-CC, l'Institut de Recerca en Salut, i IRIS-CC, i el grup de recerca de bioinformàtica i bioimatge, també de la Universitat de Vic. Bé, jo el primer que us volia dir és que la veritat és que últimament quan sentim a parlar d'intel·ligència artificial, no sé si us passa a vosaltres, però a mi és com de, oh no, un altre cop, no? Realment hi ha un abús, no? Des de l'arribada de les intel·ligències artificials tan conegudes per tots nosaltres com el ChatGPT, etc., que bàsicament estan centrades en el processament del llenguatge natural i tenen molt potencial, però també és veritat que això, que és un hype, que una mica un abús també del seu ús i a vegades arriba un moment que estem ja una mica cansats de cada dia sentir artificial aquí i allà, etc. Avui ens centrem en un tipus d'intel·ligència artificial. El que volem és, més enllà de tot aquest hype que hi ha al darrere de tot aquest tema i tot el business que hi pot haver darrere de tot aquest tema, avui ens volem centrar en tot el que s'està fent a nivell de recerca, rigorosa i realment de molt alt nivell, no recentment, sinó des de fa molts anys en aquest àmbit de la intel·ligència artificial amb aplicacions biomèdiques. I en aquest context, bàsicament, la intel·ligència artificial, en aquest context de la biomedicina, es refereix a mètodes estadístics, machine learning o computacionals per extreure informació de diferents tipus de fonts que podem trobar en el context de la biomedicina, com són les dades clíniques i dades ambientals, les dades de seqüències d'ADN, altres tipus de dades moleculars de molts nivells, que és el que anomenem les dades òmiques, també les dades d'imatge, tant dades d'imatges cel·lulars a partir de microscopia com imatges clíniques a partir de radiologia, i també molt important tot el tema del tractament de senyals, de rècords de senyals, diguéssim, de dispositius diagnòstics. Per tant, aquí al darrere, sobre tots aquests temes i tot aquest tipus de diferents dades que s'utilitzen en el context biomèdic, la Universitat de Vic i l'Institut de Recerca de l'IRIS-CC tenim experteses des de fa molts anys treballant en aquest àmbit. I avui tenim una oportunitat de conèixer això, alguns dels treballs que s'estan realitzant en aquest àmbit, i sobretot tenim l'oportunitat de conèixer-nos i establir possibles col·laboracions entre els diferents grups clínics, experimentals i computacionals que tenim a la casa. Per tant, per part meva, res més, simplement això, desitjar que disfruteu d'aquesta jornada i sobretot que aprofitem l'avinentesa per introduir i per aprofitar les discussions, que al final és el que ens farà poder trobar aquests punts de col·laboració que jo crec que són molts. I res, el que volia doncs, és, abans de passar-li la paraula al doctor Navas, doncs, donar les gràcies també a l'Institut Iris-TC doncs, per ajudar doncs, en aquesta coorganització d'aquest event, especialment amb en Jordi Villar per la iniciativa de l'organització i també per tot el que significa l'organització de l'acte. Per tant, moltes gràcies.
Buenos días a todos. Vamos a hacer trilingües al final, catalán, castellano, inglés, ¿no? Y agradeceros a todos, ¿no? el haberse registrado en esta reunión. Esta reunión tiene un origen hace varios meses, ¿no? Jordi Villar decía, bueno, deberíamos de empezar a trabajar con los distintos perfiles profesionales, es decir, la gente de computación, investigadores básicos, clínicos, seguro. En estos encuentros, pues cada uno podrá expresar dónde de sus proyectos, que se establecen relaciones y se mejoran los proyectos y sus expectativas, ¿no? Y esto, pues yo creo que fue por febrero o marzo, luego coincidió la conferencia de Valencia de un modo más general, pero quedamos en hacer esta reunión. Como ha dicho la decana, se habla mucho de inteligencia artificial, parece que va a solucionar todo. Y realmente, bueno, pues estamos en una fase que están apareciendo distintas aplicaciones y herramientas que resuelven temas concretos, ¿no? Y yo voy a compartir con vosotros una anécdota. Yo de origen soy patólogo, soy patólogo, anatomopatólogo. Y me formé en Northwestern University en Chicago. Y al llegar allí, en mi rotación por citología, tenían un proyecto muy importante y era que estaban diseñando un sistema experto, hablo del año 1974, diseñando un sistema experto en el cual introducir los criterios de malignidad de las células y incorporarlo a un microscopio de barrido, ¿no? Que hacía un screening de toda la preparación y que te decía qué células, cuántas, en qué campo pues, cumplían criterios de malignidad. Era la aplicación del concepto clásico de sistema experto. Todo esto ha evolucionado. Un investigador checo y una brasileña, Marlius Vivo y Josh Witt, los que desarrollaron este prototipo, que ha ido evolucionando y ya está comercializado. Es decir, ya está comercializado que se pueden hacer barridos de las citologías de tal manera que solo 20% de las citologías requerirán una supervisión por el experto, pero el 80% lo resuelve. Esto significa mucho, bueno, pues significa reducir al 20% el presupuesto screening que tienen que hacer los citotécnicos. Un ejemplo. Pero lo que quiero transmitir con esta experiencia es que luego cuesta madurar los prototipos, hacer que se pueda la innovación, que han pasado 50 años, ¿no? Otras cosas irán. Pero instrumentos consolidados que se implantan, la cosa no es tan sencilla, ¿no? Por tanto, estas reuniones son esenciales para este intercambio de conocimientos y de experiencias. Agradecer a Jordi Villar, que es el que pues ha estado al tanto de organizar las mesas, conferenciantes, etcétera. También pues, el IRIS-TC ha dado soporte desde el punto de vista logístico para que esta reunión sea posible. Y esperamos, ¿no?, que después de la reunión los expertos en computación, los básicos, los clínicos, hayan adquirido algunas relaciones y herramientas que les van a progresar en su carrera de investigador. Por tanto, que sea un éxito la reunión, que incorporemos nuevos conocimientos y relaciones a toda la estructura personal y adelante. Yo ahora le paso la palabra a Jordi Villa, que es el maestro de ceremonias, que será el que organizará todas las mesas redondas y la intervención de los conferenciantes.
Okay, so let's change to the third language. No? Thank you very much to both Malocalle and Professor Navas, Professor Malocalle and Professor Navas, for this introduction. I think that it's clear that what we want to achieve here is that we have interaction. Okay, so the goal for today is to achieve that interaction. In order to do so, we have organized the workshop first in just 3 hours, okay, because everybody's very busy, okay? So, and the goal is that the talks are going to be very short. We'll have 3 blocks. The first one, as I was mentioned before, is going to be about omics data analysis. The second one is going to be more general about image and in general data analysis. And the third one is going to be specifically on one topic, which is audiology and the use of machine learning techniques in audiology, okay? So it's very nice because you have from very basic research to clinical research, basically. So we have a lot of interesting stuff today to hear. The 3 blocks are going to be divided in several small talks, as I mentioned. After each talk, we are not going to have time for questions because the goal is that at the end of each block, we'll have a sort of roundtable, not a roundtable. So everybody is also, all the speakers are going to be here with the chair. And then the idea is that there is a dialogue between us and with the audience. Okay, so it's a friendship meeting. So feel free to ask, to interrupt, to do whatever you think that is interesting to learn. Okay, so again, the goal is that we know each other because sometimes we don't know what we are doing, and we do a lot of good stuff in this university and in the IDCC. So just stopping here. After the first block, we are going to go upstairs for a very small and cheap coffee break. You know, it's important to be cheap in this time of events, and then we will also have in the same place some posters that some PhD students and some other researchers in the university have put in the wall. So the goal is that, well, let's go there, let's talk, let's interact, let's learn, and let's question and answer. Okay? Okay, so first chair is going to be Professor Malocalle. Malo, whenever you.
Okay. So first block. Okay. So here in this first block, about omics analysis, the first speaker is Tomàs Montserrat. He's associate professor at our faculty, at the Faculty of Science, Technology and Engineering, and also biotechnique at the CENAC, at the National Center for Genomic Analysis. So, thank you.
I suppose the presentations are here. Yeah. Okay. Jordi, com puc posar la presentació? No. Okay. Okay. Hello, everybody. My name is Tomàs Montserrat Ayuso. I am part of the functional genomics team at CENAC. The functional genomics team is led by Ana Esteve Codina. And today I am going to present you our work entitled Revealing the Prevalence of Suboptimal Cells and Organs in Reference Cell Atlases, an Imperative for Enhanced Quality Control. So you can find this work in bioRxiv, and it was accepted last week, so I'm very happy in BNC Genomics. So in a few days, you will be able to watch it and to read it in the webpage in the BMC Genomics. Okay, so first of all, I would like to put you into context in the typical single-cell RNA-seq analysis workflow. Just briefly, normally we start with the FASTQ files, then we map the reads against the reference genome, and then we quantify the gene expression. Okay, after that, we perform a quality control in order to ensure that only good quality cells are used in downstream analysis. And then with the remaining cells, we proceed with the data processing, downstream analysis, and everything of interest. So let me focus in the quality control step. In the quality control step, normally we focus in 3 variables: mitochondrial percentage, how many reads the cells are mapped against the mitochondrial genome, the number of total reads or UMIs, and the total number of detected genes. These variables are assessed per cell, okay? And then the assumption is that that cells with high mitochondrial percentage, along with low total number of UMIs or genes, normally represents broken cells, okay? Possibly broken cells. I don't know if the micro is working. Yeah, okay. The second assumption is that cells with high total number of UMIs and detected genes, they possibly represent duplets. That are 2 cells that are counted as 1. So they are not single cells. There are 2 or more cells counted as 1. So the problem that I'm going to present you today is that often these 3 variables alone are not enough, especially in complex tissues, to remove all the poor quality cells in complex tissues. So the motivation of this investigation, this little research, started after the analysis of in-house liver samples. We ended up with this strange star-shaped UMAP. I don't know if you're familiarized with UMAPs, but this is something strange UMAP because of the star-shaped. Then we realized that the cells in the center of the star were depleted in MALAT1 transcript. The MALAT1 is a long non-coding RNA. It is highly expressed in almost all cell types, and it is retained in the nucleus. So the same cells that were depleted in MALAT1 transcript, they had virtually non-introns, non-intronic reads in that cells. It possibly means that the cells lack nucleus, for example. We don't know, but it's something strange. Okay, so the question that arose was, is the single-cell community aware of this massive artifact? And more importantly, publicly available data sets do suffer from this artifact? Spoiler alert, yes. Okay, so the first step in our research was to check the literature. If some author before us did check this fact, the intronic content. So yes, few authors before us already mentioned and used intronic content as a quality control metric in single-cell RNA-seq data analysis. The first one were the droplet QC paper. They proposed the use of this metric to identify empty droplets. And then the authors of QCLUS, DM, Sample QC, and the Brain Development Cell Atlas from Brown et al. In 2023, they all use this metric as a quality control. However, it's very few authors. Despite all these precedents, the use of nuclear markers as MALAT1 or intronic content is still not routinely adopted in single-cell workflows analysis, and it's not explained in tutorials or best practices. So in order to see if this artifact was present in publicly available datasets, we followed 2 approaches. The first one here called Intron's workflow. In this workflow, we started from FASTQ files in 4 different datasets. We processed these datasets from FASTQ files, but we used only the same cells that the original authors used in their publications. And then we just computed the nuclear fraction for each cell and classify the cells in empty droplets if they don't have intronic reads or very few intronic reads, and normal cells if otherwise. The second workflow here called MALAT1 workflow. Here we started with already processed data sets from 3 very big cell atlases, Tabula sapiens, Tabula muris, and this kidney cell atlas from Novella Roussel. So these datasets were already normalized, already dimensionally reduced, so they were ready to go. Importantly, the original authors from all these datasets performed the standard quality control using the 3 variables I mentioned before. So after processing everything, we just visualized and quantified the number of cells that were nuclei-free droplets, so without intronic reads. So in total, we analyzed more than 500 samples, more than 150 cell types, spanning more than 20 tissues in humans, mice, and 2 technologies, 10x Genomics and SmartSeq2. Okay, so let's move to the results. Basically, this is what we found. These are the results for the 4 FASTQ datasets that we analyzed from FASTQ files. Here we see a clear separation of two different populations of droplets. One population without intronic reads and another population with intronic reads. And then, interestingly enough, here in the U maps, you can see that the same cell type is divided in 2 different clusters, one without intronic reads and another one with intronic reads. And the same was observed. This is for the kidney, one of the kidneys datasets. This is for the other kidney dataset using SmartSeq2 technology. The same is observed. The same for the liver dataset, and the same for the retinal dataset. Here we can observe the rods. This kind of cell, this cell type here is divided into different clusters, one without introns and another one with introns. Something strange. Okay, in this violin plot here, we can see the intronic fraction distribution in each cell type annotated by the original authors. For example, here we can see that this cluster of hepatocytes, the cluster called hepatocyte 6, is mostly made up by cells without introns. In the retina dataset, we see these rod cells here with very few cells with intronic reads. And in the Tabula muris SmartSeq2 dataset, we see that these 2 cell types are made up basically of cells without intronic content. Importantly enough is that the authors already recognize, for example, this cluster as the cluster with less capture reads and unclear origin. So something happening was with this cluster. In the rod cells, these rod cells already were described by the authors as degenerated cells because of the MALAT1 depletion. However, here the authors didn't report anything. Okay. Then we wanted to be sure that MALAT1 could be used as a surrogate variable of the intronic fraction. So here visually you can check in the UMAP, this is for the retina dataset, but the same is happening in the other 3 datasets. So in the retina here, you can see that MALAT1 recapitulates very well the intronics fraction and allows us to identify the nuclei-free clusters. And here in the left, we have the separation of the cells in empty droplets, so cells without intronic fraction, the population of droplets without intronic fraction in red. And in blue, we have the rest of the cells. So the cells with intronic reads. And we can see that MALAT1 expression is very low in the red box and normal in the blue box. And statistically or mathematically, we computed the Pearson correlation coefficient with MALAT1 expression and intronics fraction, and we see it's quite high from 0.78 in liver to 0.92 in the retina dataset. Okay, so next we move to the Novella Roussel kidney dataset. Here we wanted to check that MALAT1 expression is able to inform us about the presence of nucleus or not. So here this dataset is made up of 59 samples with 2 technologies, single cell and single nucleus. So we have the ground truth that single nucleus, we have nucleus there. Okay, so in this UMAP here, we can see that the subset of the single cell subset, so the cells from the single cell technology, we have this, again, the same cell type divided in 2 clusters, one without MALAT1 or depleted MALAT1, another one with MALAT1. And this is not happening as expected in the single nuclei subset. Here we have the distribution of MALAT1 expression in both subsets. Okay, and what we saw is that the mitochondrial content, as expected in the nuclei present droplets, was moderate and very high in the rest of the cells. Then we moved to the tabula sapiens and tabula muris just to check what tissues are more affected by this artifact. And we saw that, for example, the kidney in the tabula sapiens was mostly affected by this artifact, and all the cells labeled as kidney epithelial cells were MALAT1 negative with exactly 0 counts of MALAT1. Other tissues, prostate, and heart were also affected, and the same but less extreme was seen in the tabula muris dataset. Other tissues were less affected, like the immune cells. And that's just conclusions. Basically, that main important publicly available single-cell analysis contains significant fraction of compromised cells that could be easily identified using nuclear markers, and that MALAT1 can be used as a surrogate for intrinsic content in those datasets where raw data is not available. And just points for discussion, what reflect nuclei-free, classical empty droplets, filled with amino RNA, etc. We should include the nuclear markers in our pipelines or workflows in single-cell analysis. The contamination algorithms are not enough because tabula sapiens already used the context to remove ambient RNA, and we can see the same artifact. RNA velocity analysis can be importantly biased by this factor because they use the ratio of spliced and unspliced reads, and that different dissociation techniques in the lab may yield different cell type proportions and also may affect differently to this artifact. And that's all. Here you have the reference, and thank you very much for your attention.
Thank you very much, Tomás.
Can I sit?
Yes. Our next speaker is Roger Casals. He is researcher at the IRIS-TT and a PhD student at the Computational Biochemistry and Biophysics Lab with BioSquare Group. Thank you.
Hello. So, què he de fer? Algú sap què he de fer? Això? Vale. Okay, so hi everyone and welcome, and thank you for inviting me here. Thank you for the organization. As Malu said, I'm Roger Casals. I work in the IRIS-CC in the bioinformatics and bioimaging group, where we are developing a web page which contains user-friendly transcriptomic tools, machine learning, deep learning tutorials, and integration of different omics tools. So this group is led by Jordi Villar, who's also one of my thesis supervisors, together with Adrián López García de Lomana, and I'm enrolled in the PhD Bioinformatics Inter-University program of the University of Vic. And today I'll be talking about mechanistic prediction of cell state transitions at single-cell resolution. As many of you may know, diseases, for example, like cancer, exhibit high cellular heterogeneity, and while bulk RNA-seq data failed to unveil this complexity, single-cell RNA-seq data that you may now be more familiar because Tomás explained how to explore the data. So it can capture, it can identify phenotypically cell states. So as you can see here, it can also allow us to identify dynamics, for example, from one state to another state. So, but as we know, there are around 20,000 protein coding genes. So if we want to reduce this information, we capture the regulatory mechanisms. How do we do that? We build gene regulatory networks. Those gene regulatory networks may be built by transcriptomic data or by prior knowledge data based on interactions. But we are more focused, we are interested in combining transcriptomics and ATAC-seq data, which gives us information about the chromatin accessibility. That means where the regions of the DNA are open. And we can identify those cis-regulatory elements that can be binded by a transcription factor and can module gene expression. And after that, we can, through some modeling, through some multivariate linear model, for example, we can infer the gene regulatory network, which is basically a transcription factor interaction to target gene. But what can we do? Which are the applications of those gene regulatory networks? We can do some topological analysis. We can do comparative analysis, comparing health and disease. We can infer transcription factor activity. And moreover, we can do in silico perturbations. What do I mean by that? For example, as you see here, this is a gene regulatory network. And for example, in this case, imagine that we knock out this particular transcription factor that is affecting this gene expression. So when we simulate data, we see that in this case, we only follow one path of the bifurcation. So we can assume that this particular transcription factor and the gene expression is involved in this path. But how can we link AI with gene expression? So the goal is to predict gene expression on unseen conditions using generative AI. So for example, in this case, imagine that we have a cell type that has been perturbed. It can be perturbed with a drug. And then, as you may know, drugs can alter gene regulatory networks activities and influence gene expression. So we have in the cell space, we have cell type A, we perturb, and we have its gene expression perturbed. So what happens if we have cell type B and we apply the same perturbation? So we can predict its gene expression after the perturbation. And this can be done across different biological contexts, even a species. As you can see here, from the response of phagocytes on pigs, we can predict its response in rats, for example. And this is linked to the first project that I'll present you. It was a competition hosted by Open Problems in single cell analysis, where the goal was leveraging PBMCs, try to predict gene expression on unseen conditions. So basically, we use some machine learning and deep learning to predict those changes of gene expression. And we did predict, we got on the top 10% of the competition. And something that's remarkable is that none of the first solutions were able to integrate prior information. So this means that they didn't use this multi-omic approach. They didn't use gene regulation. So they only use good models, good deep learning models to predict. But what do they are predicting? We want to understand if those black boxes can really predict biology. So the second project that we are involved on is on the analysis of single-cell RNA-seq dataset in collaboration with Vall-d'Hebron Institute of Oncology, in particular with Lara Nonell. We're leveraging single-cell RNA-seq dataset from different patients that had squamous cell carcinoma, a type of skin cancer, and were treated by immunotherapy. So we have samples before and after the treatment. And in this case, we want to assess the differences in the T-cell response of the immunotherapy treatment. So first of all, we capture the trajectory. And in this case, you can see some big differences. This is pre-treatment condition. And on the bottom, we have post-treatment condition. So for example, there's this trajectory that we can see on post-treatment condition that wasn't on pre-treatment condition. And we were highly interested in doing a comparative analysis on exhausted CD8 T cells, which is a highly heterogeneity cell state. And we compared the different subsets of T cell exhausted in the exhausted population in pre and post-treatment conditions. And we identified a novel exhausted phenotype that may contribute to positive immunotherapy outcomes. And I talked a lot about gene regulation, gene regulatory networks, multi-omic gene regulatory networks, but there's a lack of comparison. There's a lack of knowing if those methods really perform well. So this is what we're doing in collaboration with Saez Rodriguez Group, in particular with Pau Badia y Montpel. we are establishing a pipeline to compare all these multi-omic gene regulatory networks. And how are we going to evaluate them? We thought about different metrics, mechanistic, prediction, some prior knowledge metrics, and we want to know how the different methods perform in each metric. So in this case, we tested on different conditions, on different data sets, even on human and mouse, and we rank them. So if the method is on the top on each task, mean that it can perform well this task. So after that, this benchmarking on multi-omic gene regulatory network methods will highlight its strengths and limitation, and it will open future discussion in this field, because we are seeing that maybe with a prior knowledge gene regulatory network, we can achieve the same information than a multi-omic gene regulatory network. And also we are interested in incorporating those gene regulatory networks to predictive tasks. And finally, I would like to thank IDCC and my supervisors of the thesis, and also my peers, the PhD student team. So thanks for your attention. Thanks for your attention.
Speaker, David Pujal. David Pujal is a researcher, PhD student at the P Square group, and also the IBE, the Institute of
Evolutionary Biology.
Evolutionary Biology. Okay, thank you.
Does this have a points mark? Does someone know? Sí. Ah, cool. Okay, so good morning, everybody. Thanks for being here. I'll go a bit off topic compared to the previous talks, but well, as Malu said, I'm David Pujal from both the University of Vic and the Institute of Evolutionary Biology. And I will talk about the identification and functional characterization of piRNAs in Blattella germanica. So let's start by defining what are piRNAs. These are small non-coding sequences, RNAs, from 26 to 31 nucleotides in length. They are typically characterized because they are a U-biased first nucleotide at 5 prime. And well, the main function that was first attributed to piRNAs was to control transposable elements. These are moving sequences in the genome in order to preserve the genome stability and ensure the fertility of individuals. More recently, many new functions have been linked to piRNAs in humans, diseases such as cancers, in insects, which is my topic, wing development, the embryo patterning, sex determination. So lots of different processes are appearing to being regulated by those piRNAs. They can act both on the transcriptional and post-transcriptional levels by either binding to the target and producing its cleavage, or by generating epigenetic changes, such as histone modifications. Finally, they have quite a complex biogenesis. I'll not go into detail on it, but they have two pathways, the primary one and the secondary pathway, which is also called ping pong. I will be talking about this later. Mainly to generate the secondary piRNAs, they form pairs with a primary one, and they overlap 10 nucleotides. And this is what makes the ping pong signature, which we will analyze later. So I investigate this in this nice cockroach here, Blattella germanica. And we want to see how these are regulating the development of the ovary. So the ovary development is controlled mainly by two hormones, which are ecdysone and juvenile hormone. Here you have the profiles of the hormones. This is during the last instar nymph, so before being adults, and then during adults. What we did was to generate some small RNA seq data. So we will be dealing with transcriptomic data on the main hormonal peaks and changes of this development in order to analyze the dynamics of the pyrRNAs in those key shifts of this process. So the pipeline of the analysis that we did is the following. The general idea is that we start with huge amount of data and we want to narrow it down to the most relevant one. So first of all, from the FastQC files that we obtain, we remove sequencing adapters and we select only for those reads that are in the range of the pyrRNAs. This resulted in over 33 million reads. Then we align this to the Blatella germanica genome. We don't allow for any mismatch because we want to know where these pyrRNAs can be produced in the genome, so the exact sequences. And we still are left with over 22 million reads. In order to reduce the complexity of this data set, we applied a filter to remove all the lowly expressed sequences and extremely infrequent ones. And this resulted in an increase of homogeneity of replicates, which is also always nice. And we ended up with these 191,000 sequences. Finally, we collapsed 3' variants, which I'll not go into detail now, but the final set of pyrRNAs that we will analyze is this 129, which is quite an important reduction from the first 33 millions. So this is the set that we use to identify the length of these pyrRNAs, where they are produced in the genome, if they are if they have these ping-pong signatures, and some dynamics of their expression. Finally, we looked for pyrRNA pairs, so primary and secondary, that are generated in genic regions and are hopefully related to gene regulation. So going on to some of the results, here is the length, so the number of pyrRNAs according to their length. We can see that in Blatella germanica, most of them are between 28 and 29 nucleotides. And when checking for their sequences, we can see that for the primary fraction, there is this U bias that I talked about in the beginning, which is characteristic of pyrRNAs. While in the secondary fraction, apart from the U bias, we find this A bias in the 10th position. This is the ping-pong signature because since they are overlapping 10 nucleotides with the primary, and the primary almost always has a U, the 10th position of the secondary will almost always have an A to be complementary to this U. So this made us pretty confident about the identification process, and we are quite sure that these are indeed pyrRNAs. So then we proceeded to see where these are being produced in the genome. As expected, most of them are found in transposable element regions. This is the canonical function of pyrRNAs, so most of them are here. But also we find that a lot of them, well, not a lot, but considerable amount are being found in genic regions, UTRs, and coding sequences as well. Also, we see that there is a tendency to increase in the last days of the cycle of this maturation process. So we'll try to focus on this as well, because it's quite interesting to see this increase of genic pyrRNAs, which might be regulating some genes. When checking the expression, we can see that we find a cluster of all the libraries from nymphal stages and the first adult stage all together. And then there's a big leap to the last stages of the developmental cycle, which is adult day 5 and 7. This matches the definition that we have of immature and mature ovaries, which the transition would be around adult day 3. So these would be the mature ovaries, and these would be the mature ones. And when we check the differential expression between these transitions, which is this graph here, we see that for all the nymphal transitions, so when the ovary is still immature, there are no relevant changes in the expression of pyrRNAs. But when there is this shift to the mature stage, there is a major shift in the expression patterns of all the pyrRNAs. Well, not all of them, but most. And it's still, so in those last stages, there are still a lot of things going on due to pyrRNAs, because there's just a lot of the changes going on here. So finally, I'll just give you an example of some hints that we might be getting of these possible pyrRNA gene interactions that we're interested about. So this is one example of these pairs I talked before. So this is the expression of one gene. We don't know what gene it is because the annotations for Blattella germanica are not great. So we still are not sure what this is, but it's only expressed on the last day of the adult in the ovary. And this is the expression for the antisense pyrRNA, which would be the primary, and the secondary pyrRNA. Our hypothesis here is that the antisense pyrRNA is complementary to the gene, so it can bind to it and produce its cleavage. When this happens, the positive, the sense pyrRNA is going to be produced. And this, at its turn, might have some targets elsewhere, producing a network of regulation between genes and pyrRNAs. So this is what I think might be happening here. So once the gene is expressed, we have a peak of the negative, but the antisense pyrRNA, in order to cope with that, it binds to it, it may be cleaving it, and then we'd see the rise of this secondary pyrRNA. We are going to, in the near future, perform some wet lab experiments to try to validate these interactions and see the specific functions of this. So that was all. Thank you very much for your attention. And
I invite you, the speakers. Yeah, to be here. Em poso aquí, que tinc la... Okay, so thank you very much for your presentations. Now we have some minutes for discussion. I would suggest that this discussion is not technical, since the presentations were already technical and perhaps hard to follow for the audience. But now I think that it's interesting that we discuss on the on more general terms. Two of the presentations were about single cell analysis, the other about pRNA. So maybe we can start by discussing on or commenting on the importance of single cell analysis. And this technology has extended the analysis of gene expression, mRNA data. So what do you think about, and also the audience, about the importance of this technology and this analysis?
I think that what a single cell RNA brings to us is the power of resolution. So we not only see the transcriptomic analysis at the bulk level, so we can so on specific cell types in its tissue. And for example, the analysis performed by our colleague, maybe at cell resolution, to bring much many more sites. I don't know, the VNAs. I don't know, maybe it's some cell type that is expressing this pyrRNA. No, because we don't have the power of the single cell. We lack this information.
for sure, it would be really nice to have this information, but at the same time, I think like the more you know about the organism, the more use you can have of these techniques. So like in humans, for example, everything is quite well annotated and known. So it's easier to go deeper into whatever this single cell sequencing can tell you. In other cases, I'm afraid that maybe like you would have a lot of information that you could not be able to read because of lack of basic knowledge on the organism or on the genes. So maybe it's a process that it's a really good tool once you get to a certain point of knowledge. And I don't know, I'm just thinking out loud.
Roger?
Yeah, I suppose, as you said, it's a process because I think in human, for example, now they are applying not only even single cell, they are applying multi-omic approaches with spatial, with chromatin accessibility, with phosphoproteomics. So like there are a lot of people doing a lot of things. And for example, I don't know if it's in your case, but no, not many people will be working on single cell, for example, to this cockroach. So this is something that we should take into account, that we should like the balance would be, I don't know, balanced.
When we see the numbers of the single cell analysis, we see many large data sets, but at the individual level, I mean, the number of cells is very large, but what is the variability between individuals in this analysis? Is this another issue? Like, it's perhaps needed an hierarchical process that we analyze the problem at the cellular level, but then we combine to the individual or to the population?
I think that's a difficult question because, as probably Thomas may know, labeling the cells, I think it's very difficult. And also when the cells come from different individuals, this also complicates more the task of labeling and integrating the things, because you have to have a prior knowledge in order to label those things. But there's huge variability on individuals, on cell types, on even within a cell type. So that's a difficult task that has also to be approached.
Well, yeah, it is true that some years before, I mean, 2 or 3 years ago, researchers didn't perform replicates in single cell, and the most common thing was, oh, yes, I have one data set of single cell, and we want to perform differential expression analysis. And this was a limitation, of course, because you couldn't estimate the variability between people. But I think that nowadays people are sequencing more and more, and at least in my projects now, I am starting to have replicates, even inclusive many replicates. I have a project where I have I don't know, like 10 replicates, biological replicates. So this is an issue that we are solving in the field. But it is true that with with increasing the number of replicates, now we have new problems like handling batch effects. That we have a lot of batch effects in single cell. I always say that in single cell, it's like the experimental design. It's something. Well, maybe it's not as important like in other topics. I'm not saying it is not important, but sometimes you get terrific experimental designs, and I think this is a thing that the field should take into consideration, at least in the future. Do you want to say something about single cell in cockroaches? No?
Is there any comment from the audience?
You already mentioned this, no? That the fact that that analysis, the current data analysis, is based on what we have, but the perspectives of what we can have are huge, no? And in that sense, do you think that research in general, because you are reading a lot of papers, you read much more papers than your supervisors right now, okay? So you read a lot, you know a lot. So do you think that, do you feel this bias of saying, well, because we have 10x and we can do single cell or we can do pRNA analysis, and then that's the most important thing ever in the world, no? And I forget about the rest, no? Do you think that you see this bias in the sense that technology is prioritized before biology in that sense, or not? Or you see that people is really going from top down, no? Like say, okay, we have this biological problem and then we are going to tackle what are the tools that we need to use.
Well, for sure there are trends, I guess, and at some point there's a new technique and everyone wants to do the technique, maybe if it's not strictly necessary. But it's also something good to explore new technologies when they appear. And yeah, like this bias, where I feel it most in my case is on the organism level. So there are organisms entire families or clades that we know almost nothing in terms of genomic and proteomic information. Whereas in other species, I'm really jealous when I see people or articles on humans or mouse. It's like, oh, it's so nice to have everything labeled and annotated. So I think this is something that we should, community, try to address, and because you never know where the next really big, discovery can happen. It might be on this cockroach, who knows? So try not to forget the biodiversity that we have and try to explore as many organisms as possible. I think that should be good and should be done. But in terms of techniques, yeah, there is also these trends, I guess. I don't know.
Well, in Senac we have a team about annotation, so maybe you can talk with them. They annotate many things, many animals and plants. But I think yes, that bias exists. For example, but I think it's not because of the research. Sometimes it's because of the pressures in the industry, because if you are using the top technology, you are going to reach easily publications in top level journals, that if you are using good technologies but they are not the top, it's more difficult for you to access to these kind of journals. And I can say that normally in my work, in my everyday work, I see that people, there's a new single cell dataset to analyze, and there's no hypothesis, because sometimes people use single cell as a hypothesis generator. And they say, because normally I ask, okay, what's the hypothesis? And the answer normally is, no, it's just to see what appears there, what show up. So well, that's good and it's not good. The good thing is that, okay, we are using, the good thing is that I have a job. And then another good thing is that, okay, we generate a lot of information, a lot of results, and maybe this generates more hypotheses and then more work for me again. Yeah, that's true, but that's the bias. And I think the bias is more top down, more first of all technology, and then let's see what excuse I have to answer some biological question, and then I can publish in top journal.
Yeah, I agree completely that the technology indeed is driving all the research in this field, and then we find the use of this technology, and sometimes it usually is very relevant. Okay. Yes.
Hello. Yes. Good morning. Thank you first to the three of you for the talks. I'm not in the field, but I'm understanding that this event is flexible enough to be allowed to ask stupid questions. So I'm gonna ask the first stupid question of the day. If I got what you were explaining more or less right, all the three of you have huge amounts of data and you want to somehow reduce it to a fewer parameters, right? This is a common. But I noticed that the two single cell ones use a UMAP, which is a nonlinear algorithm. And that you are not the single cell one, you use a PCA, which is a linear algorithm. What makes in your field to decide which algorithm to use? If it's a nonlinear algorithm that also exploits local structure in the data, or a linear global algorithm?
This is dangerous because our chair perhaps wants to say something also.
Okay, in single cell, well, in RNA-seq, normal RNA-seq, bulk RNA-seq, we do use PCA as our colleague, but in single cell, we also use PCA, but this is an intermediate state, because normally what we see in single cell, if we have several cell types in the same sample, the PCA, this dimensionality reduction technique, is not enough to separate all the cell types, the different cell types. And UMAP is, we use UMAP from the PCA dimension, but it's only used as a visualization technique. So all, for example, clustering is used, we perform clustering on PCA dimensions, and then we overlap this clustering in PCA dimension in the UMAP. But it's just as a visualization technique, because we see the two dimensions of the PCA, it's not enough to segregate all the cell types in our sample. So it's just a matter. It's visualization. It doesn't have any other meaning, this UMAP. I don't know if I have answered your question.
Okay, thank you. I think we should stop here the discussion. So thank you very much again, and congratulations for your work. Thank you.
Okay, so now we have this coffee break upstairs. Okay, so you have to go down and then go up, and we'll be here in half an hour or so.
Sí, endavant, hi everybody. I want to present.
Hi. Okay. Okay, so let's start the second section of this event. We have two talks in this part. The first speaker couldn't come. The first one is Pere Martí from the DSP group, and he will talk about the time series processing and some practical applications of it.
Well, thank you very much. Then I want to present. Well, the presentation, the outline of this presentation is to give some basic information about time series, the use and goals. I explain a little bit about different time series models, and I select 3 applications. All of them are quite different between them, and shares the fact that has a lot small of data sets labeled. So, well, what a time series is? It's, as we can see, are sequences of data points collected and recorded at specific time intervals. What we can see in the left picture is a data series represented as a function in which we can observe different patterns inside. For example, we have cyclicities, we have a trend. The cyclicity could be observed in different levels, etc. But what we usually have in more interesting problems is not a single data sequence. We have a collection of them. And what are interesting to observe is the patterns that are observed not in a single one, patterns that are represented in the several data series. The same could be observed in frames images of a video, for example, tracking objects or something like this. And in a more bigger problems, we can organize information on, for example, tensors. And this is more challenging to observe relations inside the tensor, but this could be done in bigger problems. So, well, we want in a data sequence first to observe such patterns, such relations inside the sequences. Then we want to understand what causes such patterns. Well, it's a high level of the knowledge, and if we can, to forecast the future values. And ideally, given such information and the predictions done, what we want is to suggest optimal actions, sometimes based on such predictions. Well, in order to deal with time series models, we can divide as a big division different approaches. The classical approach, which is mainly based on statistics, well, all is based on statistics, but the first one is the moving average, autoregressive moving average, well, a big family of such kind of approaches. But what we are interested here is in machine learning models, because we have observed that in many problems these approaches overcome the previous ones, the classical models. And here we have huge families of models, very, very huge. Well, so dealing with machine learning models, there are different steps to fulfill. The preprocessing is a very important one, but what is crucial in order to obtain good results is to have a precisely labeled data set. More or less, if we have a good labeled data set, we can reach good results. If this step is not fulfilled, then it's difficult to obtain nice results. And another important step is what we sometimes know as feature engineering, which it's very important for small data sets. For example, it's the way in which we represent the information appropriately. For example, if we work with voice, we need to compress information in an appropriate way that brings the information required. And here, our group has some expertise because our background is in signal processing, and it is maybe our strength in the group. And here I select these three examples. And in this application, the time sequence are obtained from drawings. We use a tablet, and we record the process of the drawing. We can measure the coordinates, we can measure the pressure, we can measure the angles, the time in which the pencil touches the surface, the time which is in the air, and so on. And we can extract a lot of information from writing datasets, handwriting datasets, or drawings, in this case a house, or there are some tests that are related with drawing an Archimedes spiral over a paper that is used to monitorize cognitive impairments over time, and especially in a disease which is called essential tremor. And well, here we have the typical architectures we use to classify such images, such drawings. I don't deep in. Another kind of application, very different, is when we try to predict some events. No? For example, in this case, the application is thought to work in a mobile application in which people who have non-suicidal self-injury tendencies, especially young people, interact with an application and monitorize his state, emotional state, and some information. And based on this information and an historical we have previously, we try to predict what such event could happen, and then we return some advice and some actions to do. What is difficult in such kind of applications is that the data set is small. We have a few people, very documented, and the data set is very unbalanced. There are people that are involved in this application, but others not, really not. In this kind, we use very simple models, 3 models, that if we can model the process in small trees, these models provide information about what is the process that triggers the response. And finally, we have selected another kind of application in which the time series in this case is recorded acoustically. And in this application, we try to segment the signal in inspirations, pause, and expirations. And this seems easy, but it's not, because there are sound around, there are so many difficulties. The people could breathe by the nose, by the mouth, combine it, very fast, very slow. We have a lot of combinations. And in this case, what is relevant to solve good this application is to apply a good features to represent the process. And well, this all. Thank you for your attention.
Thank you, Pere, for the presentation and for sticking to the time. That was perfect. Thank you. And the next speaker is Laura Lempere. She's from the By2 group, and the title of the talk is going to be Decoding the Impact of Aging in the Interaction Between Working Memory and Visual Attention.
Okay. Hello, good morning to everyone. And yeah, I will be talking about decoding the impact of aging in this interaction between visual attention and working memory. And I will be tackling this aspect, trying to relate it to artificial intelligence, but in a very broad sense. So I'm not going to be specifically talking about specific classification techniques. I will just walk through different modeling and analysis techniques, and I will just bring a few of these topics into it. So first of all, we need to understand what working memory is. And working memory is a cognitive function, and it is the cognitive function which allows us to maintain and manipulate information for very short periods. So this is the type of cognitive process that we use when, for instance, we are given a phone number and we need to remember it just prior to actually taking note of it. It is a very important cognitive function. It is at the basis of intelligence, and in fact, it is impaired in many neurological conditions. Something which is known, it is one of the most studied cognitive functions, in fact. Something that we know about working memory is that it's got a limited capacity. And this, in order to understand it, I've just put a small sketch in there. And in here, what we can see is that, and let me see if I can use this. Yeah. What we can see in here is that this is a typical test used in experiments, and we normally show a few items to our experimental subjects, and this change in number. And we show it for a very short period, then we take them out and we ask the subjects whether this particular object was present in the stimulus set. So it is this idea that we no longer have the information. And the amount of objects that we can keep, in fact, is limited. And typically for visual stimuli is in the range of 4 to 5 items. This capacity, in fact, depends on the task difficulty. What you have in here is a set of colors, but colors are very easy stimuli. If we change that for instance, Japanese characters, we realize that it takes a lot more effort to remember them. It is known to decline with age, and that's something that we will investigate in this talk. And something that I also want to put emphasis is that it is a temporal process, and in this experimental test, it is very clear. There is a first stage, which is the encoding one. Then we have the memorization stage, which is the maintenance of the stimuli in memory, and finally the retrieval. And what we claim is that the encoding stage is extremely important, but most of the research has been done so far in the maintenance stage. The way we tackle this investigation, in fact, is something that I want to insist, because it is a combination of both modeling and analysis. Our modeling informs the way we actually do the analysis, and the analysis informs the way we are going to be modeling our data. And this is done from two perspectives, computational neuroscience and statistics/machine learning. Typically, most of the research that has been conducted in this area considers either a simultaneous display of stimuli, so all of them are presented at once, or a sequential display of a stimuli, so they are displayed in sequence. But there is very few research which considers both simultaneously. And this is something that we have tried to tackle in the research. From the computational perspective, we use neural networks, but these are not artificial neural networks. These are biophysically plausible models, which basically model the potential membrane of the neurons. It uses an electric model of that, and it is relatively realistic in that it uses synaptic currents of both excitatory and inhibitory nature. And this type of models, I will be very brief, but basically what they consider is a pool of neurons. We typically work with 10,000 neurons. And let's assume that, for instance, we have 10 different stimuli, 10 different colors. And we have 10 different pools, which are selected to each of these colors, okay? And these networks have a structure, they are recurrent networks, so all the pools within, all the neurons within the same pool are connected very strongly. This has been shaped by Hebbian learning. And the connections between different pools are weaker. All of the excitatory neurons are connected to the inhibitory neurons, which in fact inhibit the activity of all of the neurons. So this set a dynamical system, which is quite complex. And in fact, this type of models, which are attractor networks, can be used to analyze or to study decision-making, working memory, different cognitive functions. It depends the regime in which you set the system to work, you will be modeling one type or other type of data or processes. And something that we brought into these models, which were known for long, is synaptic facilitation. And this basically means that we include some calcium accumulation in the presynaptic terminals. And this means that the neurons which have received excitation at some point will be more prone to respond if they receive excitation later on. So with this type of models, we are able to model the activity of our reduced system, which in this case is the working memory system. There are different approaches to study working memory, but the most prevalent one is assuming that a neural correlate of working memory is what it's called persistent activity. So it's when we have neuronal pools which persistently respond highly in the period in which we don't have a stimulation. This is the period where we have a stimulation. This is when we don't have the stimulation, and this is when we test. Okay? So as I said, when you work with this type of modeling, you need to set the parameters to work in the regime that you are interested in. And this value for this particular parameter of the synaptic facilitation has been widely used in the literature. And as we can see, when we use it, we can actually keep, if we are stimulating 9 stimuli in here, 9 pulses, we can see that on average, we will be keeping 4 or 5 items, which is what we want. This is what experimentally has been shown to be. So in our previous papers a few years ago, we used these values and we reproduced experimental results. We were very happy, but then we made this what if question. And the what if question is, what if instead of showing the data simultaneously, we show the stimuli sequentially? Does this model work? Because if it doesn't, we are trying to model the brain. So if it doesn't, we need to change the model. And we need to change the model. If you use this tau f, which was the one that we were considering, it is true that we keep, again, here we are stimulating sequentially. So each of these gray shades means that we are first stimulating the first stimuli, second, third, fourth, fifth. So in here we have the delay period, the one in which we maintain. So yeah, we do keep 4 or 5 stimuli, and that's fine. But the problem is that when you show the data sequentially, serial effects might appear, and it has been reported in the literature that we tend to remember best those items which are shown later in the sequence. But in this case, these guys don't even have the chance to access the memory. And this is because while we were exciting the network, we were recruiting a lot of inhibition, and they don't have this chance. That's why we actually say it is encoding, which is very important, and we need to look at that. So we started to explore the parameter space, and we saw that we can reproduce both an appropriate number of data being kept and also the so-called recency effect, which is we will remember better those items which are shown later in the sequence. So we put our system to work in this regime. You can see here what happens when we change this parameter. If we use the traditional one, the likelihood of keeping the first ones is very high, and then the likelihood of keeping the last ones is very low. But as we change that, we modulate this curve. If we want to keep on average 4, 5, we are best off if we work in this range, okay? So this is what we did, and we ran our models. In fact, this doesn't only depend on this tau f, and this is something that we claim in previous works. It depends a lot on how you stimulate. If you stimulate for less time, you will be keeping less stimuli because you don't have so much excitation in the brain, and also on the amount of stimulation that you receive. That's why I said if you show colors, it is relatively easy because we have a lot of neurons which are tuned to respond to those colors. If you show kanjis, you have far less neurons which are tuned to that, and therefore you need a lot more time or a lot more stimulation. So if we want to work in an appropriate regime, we are going to be working over here. So we set the model to work in there, and these were the predictions. We know that the capacity of the working memory is about 4, 5. So if we only stimulate 1, both under sequential or simultaneous conditions, we keep 1. Same for 2, same for 3. For 4, we start to have a little bit more problem. And for 5, when we are a bit over our capacity, we actually tend to remember better the last one at the cost of not remembering so well the previous ones. Same for 6, 7, 8, and 9. Okay, so we really very clearly have this recency effect, and that's fine, and the model in this sense works. But we found something which was very interesting, and that's where I was trying to go. And it is the fact that there is a collapse of working memory when you stimulate overcapacity, okay? So again, this is the amount of items that you keep. If you stimulate 1, you keep 1, 2, 2, 3, 4. For 4, sometimes you keep 4, sometimes you keep 3. For 5, sometimes you keep 3, sometimes you keep 4. But note what happens for the simultaneous condition when you show 9. The amount of items that you keep is very, very low. So this means that there is a collapse of the working memory. And this guided our data analysis when we came to analyzing the experiments, okay? So just keep in mind that, because I will go back with experimental analysis. And now we just put the other hat, which is the data analysis. We run experiments with the experimental paradigm that I just showed to you in the simultaneous condition. And we had 90 subjects, and we are very grateful to them because they were very, very nice to come and do a task which was hard. We actually balance the ages of our subjects. So we have 30 young adults, 30 middle, and 30 old adults. And I know that the range can be, I mean, you can discuss that, but that's it. And we had 252 trials divided in 3 blocks, just not to exhaust our experimental subjects. And we perform a battery of neuropsychological tests, like 1 minute. Wow, I need to be fast, very fast. So we ran this battery of neuropsychological tests, 16 tests, to ensure that they were healthy, and we monitored their activity, the performance with eye tracking. Okay, so the results that we got were something like this. The task is performed at a very high level, so both true positives and true negatives are very high at the level of 85, and the incorrect responses were really low. And something that I noted in here that I've noted for you is that the specificity and sensitivity changes between old and young people, okay? And specifically the sensitivity for the old population is worse, which means that they actually encode worse the true positives, so they might have problems to encode, and this is what was guiding our research. If we consider in our experimental test, we show either 2, 4, or 6 items. And what we can see is that performance decreases with the difficulty of the task, and also it decreases with the age. And this reversal that I was mentioning, in fact, happens mostly when we consider 6 items. So it happens for difficult tasks. We monitor the reaction time, so the time that people try. I will need 2 more minutes. I'm sorry. The reaction time is the time that people need to press a yes/no button. Okay, so there is a motor action in there, and we can see that the reaction time also includes this idea that more difficult tasks need more time, and also the reaction time increases with age. Note also that, of course, this is 2, 4, 6, so the shape of the density distribution changes, but not so much for young people as it does for middle and old people. So very easy tasks has performed relatively well for the old groups, but they are very, very different from the 4, 6 set size, which are the difficult tasks. If we divide the analysis by true positives, true negatives, false positives, false negatives, what we can see is that there is a very clear distinction between correct responses and incorrect responses. And as time goes by, with aging, this difference fades away. So this is also something which is quite important. And something which is remarkable also is that the true responses, either positive or negative, that they are sorted. Okay, so true positives take longer, shorter reaction times than true negatives, than false positives and false negatives. So there is some processing which is going there, which means that although you are giving a response of this item is not in the set, you know something because you take longer in order to reply that. And that is something which needs to be investigated a lot. In order to analyze this data, we have used, and this is where machine learning, if you wish, comes into play, or statistics. We use a multivariate logistic regression analysis, and we did it by using odds ratios. So basically, assessing the probability of committing an error when a factor is present as opposed to when it's not present. And this allows us to define, because we are modeling error, risk factors or protective factors. And what we can see is that age, clearly, as we age, it is a risk factor. Young people versus middle people, it is a protective factor. When task difficulty increases, this is a risk factor. It actually increases by 8-fold. And also the reaction time becomes a risk factor. If we assess the capacity of of the system, the working memory capacity, this is a probabilistic model in order to, from the behavioral data, estimate the capacity that we have. Here we find the collapse. If we show 2 items, for all the populations, we have more or less the same reply, and all of them keep more or less the same number of items. For young people, if we show 4 or 6, this increases. For middle-aged people, this increases, but not so much. And for the old population, this collapses. And this is something which wasn't available yet in the literature. In fact, if we use this data not in groups, but as a continuous variable, we can see that this is the trend for 2. So the capacity doesn't change along the years. For 2, there is a slight negative slope, but for 6, this slope is much larger. And the point in which they cannot be distinguished, in fact, for this particular data, starts at 55 years old. Another type of analysis that we did, multivariate regression analysis, to analyze how age and set size depend or contribute to the estimation of K. All of them are very significant. And in fact, also the interactions, the interferences between the variables are very significant. And now I finish with that, and I just speed up a little bit. Since we are very interested in the encoding part, we are monitoring the encoding part with site tracking. So we have defined different types of searches, which actually change gradually from a lot of focus on the stimuli to focus more distributed between stimuli and background, only background, and very, very peripheral, okay? Preliminary results tell us that young people tend to scrutinize a lot the items, and this correlates a lot with well behavior. So it is the search strategy which changes and which actually has an impact on the capacity. And just finally, we have used also manifold embedding techniques. You have talked about PCA. It is very similar, but more general because it's not only linear. And this comes from the analysis of the 16 neurological tests. We have there a lot of dimensions, but it is very difficult to assess the 16 dimensions at once. So we have performed this manifold embedding in order to agglutinate this cognitive variable. And we can see that they actually agglutinate very well. And just one dimension would be very, very interesting to analyze. And in here, although it is true that working memory collapses and declines with age, there are people who are super agers, because here we have a person who maps in this part of the space where you should have young people. So this is a technique that we hope it will be useful to track healthy aging and to actually monitor interventions of cognitive interventions. And that is it. But the most important part, this was not my own work only. I have contributed, I mean, this is part of the PhD of Marta Balaguer, and we have worked together with Marina Martinez for Universitat Jaume I. Thank you.
You can stay. Thank you for rushing. That was the longest minute ever. but thanks a lot. That was a very, very interesting talk. Okay, so let's yeah, you can take some water. Let's start the discussion. I mean, the topics are kind of similar, but a little bit different as well. So from time series to behavioral like data and also more like cellular modeling and biophysical models. So it's sometimes difficult to make a common discussion. But if maybe we can start with the audience. If anyone has a question that you want to address to the speakers, more general or also more specific. Yep, Joanna.
So I have a practical question about your presentation and about computational models that you used. I was wondering, in practical sense, how do you use these models? Is this like a software package that you have to, or is it that you can, and is it like a code that you develop, for example, in your lab, or is it something that is let's say unified across the computational neuroscience field? Just trying to imagine how to work with such models. So then the second question is to the first presentation. And I was wondering what type of features you use for segmentation of the briefing sound. This looks very interesting, I think, yes. And I was just interested, what are those audio features? Yeah, I think that's enough.
Just have one microphone.
If you want to.
Thank you for the question. So the models themselves have been published. The original model comes from a paper in 2001. It is an attractor neural network. And I was working in a lab in Barcelona in which there were several PhD theses going on, and they developed the main code, okay? So we extended this, it was a single item working memory model. So we extended it to multiple items. And then whenever we want to add a new biophysical process, we just change. But it is a code in C++ and it's not publicly available. But yeah, that is basically it. Yeah.
Well, related to the segmentation, no? In this particular application, we test maybe 40 or 5 characteristics, try to observe which one works better. But very easy ones, as we segment the acoustics in small parts that are more or less stationary. In voice, for example, this time is about 20, 30 milliseconds. Here is a little bigger, the time, but we represented such frames in mean, the standard deviation, the cosinus transform, we select coefficients, the same for the Fourier coefficients, kurtosis. Well, we analyze a lot of and we try on together ones different. But an interesting experiment we did in that when it happens, it was that we sensor temperature inside a mask, and with the temperature is in the mask is very relevant, because we cool the process, then when it in expiration the temperature increase, and this process could be measured. Then combine it, the effectiveness was great. It's fantastic. So we need sometimes to combine more signals in order to work together.
Thank you. Is there any other question? Any?
Thank you for your presentations. I enjoyed it. And Dr. Laura Dembele, I have a question about your presentations. You mentioned about the kanji, like the Japanese characters as well. So that makes me wonder if you consider about demographic factors, like what are the populations you include in your study. That might cause some differences, say if some of the people, they are already exposed to understanding of it, they might have a different performance in the experiment. And also you mentioned about one outlier in the age, like middle age. I don't really remember that. So I was wondering, like, if you have also considered about the profession factors as well, would that change the patterns as well? I suppose maybe some Nobel Prize, they have better performance compared to young people, right? Yeah, I just really find it very intriguing, and it would be great if you could share a bit more about the conclusions and etc. Thank you.
Somebody else?
My question is related to genes one. It's a more general question, but how do you deal with class imbalance? Because I suppose in classification problems, this is something to take into account. So from a naive perspective, how are your approaches on class imbalance?
Thank you for the questions. Yeah, you're totally right that it depends on demographic. In the end, when I mention kanjis, because most of us are European, and therefore to us, kanjis are very difficult to encode, but it has to do with the facility of encoding, okay? So if you're very acquainted with a particular stimulus, fine. For you, it's going to be very easy because you have developed through your own evolution a lot of neurons which are responsive to that. So totally agree. Even within the same population, if you are, I don't know, if you are an expert in classification of birds, and if I show you birds, you are going to be much better than myself. I mean, to me, they are just birds. I mean, I have no idea about them. That is the first one. And the second, sorry, what was that? Sure. Yeah, absolutely. And that's a very interesting question. Absolutely, and this is why I show the slide. In fact, there is a notion which is cognitive reserve, and this cognitive reserve, in fact, can be nurtured through life. So one thing that we wanted to show was, can we propose a method in which we can monitor how people age? Because one of the aspects which are nowadays discussed is whether you can nurture this cognitive reserve in such a way that it doesn't stop all the aging, but at least it can balance it a little bit more. And surely people who have had a lot more cognitive activity, if they don't have any physical factors which bring them into disease, are going to perform much, much better. In fact, something that we have seen is that our subjects change in sociological background and in level of studies. And in fact, there is a change in that also. So we have identified the superager, and this superager, in this case, was a person with a very rich cognitive activity. Okay, so definitely. So one of the possible applications is precisely, are these cognitive training, not algorithm, these cognitive training practices which are nowadays so common, or at least they are so fashionable, do they work at all? Can we actually propose some pseudo-scientific methods to monitor that? So yes, we can. And regarding your question, I mean, I'm happy because we actually balance our data a lot. We did a lot of effort to have our data balance. But in other projects which we don't have them, we just augment the data artificially. We create in order to balance, because we know that otherwise pattern recognition methods are going to fail. They're going to optimize, or they're going to maximize performance. And therefore, if you have unbalanced groups, you will be taking a default answer. Yeah.
Yes, related to data sets. It's a very common problem when we extract real cases, no? The data from real cases. And yes, the method is to try to increase your database in the classes that are less represented, constructing artificial data. And this could be very carefully, because we can change the problem if it's not properly done. And there are a lot of techniques to create artificial data based on the data you want to represent, sharing the same statistics and, well, for example, super vector empirical model decompositions and sharing the modes. There are a lot of different things. Even transform the data and combine some coefficients between in order to create new data and reconstruct the signal, for example. Many more.
Yeah, thank you. Thank you very much to both speakers for the talks, and we'll move on.
Okay, now we switch.
Yes,
okay, so we switch roles now. Yeah. So he's going to be the speaker, I'm going to be the chair. So in this last session, actually, thank you for the timing, because actually even Laura was trying to destroy the timing, but but we are good still. So we have now 2 more talks, and they are going to be around sound analysis. Okay. The first speaker is Gerard Encina Llamas, and he's actually the dean of the audiology degree here in this faculty, and he's going to talk about towards precision audiology. Okay, but before that, he asked me if he could do some explanation about the audiology degree, and actually he has 2 extra minutes, not more than 2 extra minutes, Gerard, 2 extra minutes to present your degree and who you are.
Exactly.
Okay.
Oh, no. So yes, thank you and good morning, everybody. Yeah, I asked for a couple of 3 more minutes to present our new activities because we just, I mean, this is the title of the talk, but I wanted to say you before that we started last year in September 2023, the first ever bachelor degree in general audiology here at the UCC. And we have started the second year now in September. These are our facilities. So we have the audio lab funded by the William Demand Foundation. I was living in Denmark for 12 years, and we have a good connection with Danish foundations, and they help us to establish that. And we've done many activities. So we teach audiology. We teach students mainly like audiological aspects, but audiologists also are involved in vestibular assessment. This is what you see in the other pictures. And we've done many also fundamental lectures on basic physics of sound, couple oscillators, waves, some neuroscience. Here we were measuring the other day single units in similar animals that somebody was using in the morning. So many aspects. And we have also established, at least formally, a research group that we call Grup de Recerca en Ciències de l'Audició i l'Equilibri a Catalunya. And for now, it's empty. It's just a box. But there are some projects that we are starting that will eventually fall in that group. I can just mention briefly some of them. So we are working on establishing audiological data sets in hospitals in order to have all the clinical data that the hospital generate every day, have them organized, because nowadays we cannot use any of these tools in the data that is generated simply because it's not well stored. All the data is digital, but you cannot do anything because it's not properly stored in a database. We are planning a screening program for the elderly, from 60 or something here in the region. We are about to start a PhD, an industrial PhD with Eurecat and Amplifon on spatial hearing and diagnostics and rehabilitation and so on. We are helping the doctors at the Germans Trias i Pujol on some vestibular schwannoma toxicity project. And I'm also involved in another project with Pompeu Fabra and DTU, that's the Technical University of Denmark, and Chu Hospital, a Danish one, and Sant Joan de Déu, on using geometrical deep learning to predict the growth trajectories of ear canals in babies. So we think that a lot of activity is going to happen, and we just wanted to share that with all of you. Now we go to my thing, where I'm going to explain 2 projects in this line of precision audiology. So today, the way we assess hearing is by measuring different tests, psychoacoustical tests, or some electrical, or electrophysiological recordings, or something called autoacoustic emission, that is sound that the ear produces, and we can measure in the ear canal. But these tell us whether there is a problem or not. But those tests are not able to tell us what's the problem, which cells are going wrong in the cochlea, and which explains that deficit. And this is, we think it's going to be interesting because we have the perspective that new pharmacological intervention will appear. And when this will happen, we need to know what we need to repair. So I want to show, explain briefly 2 projects. One that we use evoke responses. This is like electrical activity in the far field from electrodes in the scalp that we can measure from neurons when we stimulate with sound. This is the postdoc project from Mickey at DTU in Denmark. And the other one, which is my project, that is looking at a large data set from a Danish hospital that they have the data properly organized and stored since 1995. So let's start with the first one. So we have good models, like signal processing models, that can account for all the transformation that the sound encounter from the stimulus to the filtering of the middle ear, the vibrations in the cochlea, the role of inner hair cells, outer hair cells, and the response in the nerves, in the auditory nerve. And from this, we can have simulation, and those models are pretty good. We can have simulations of the compound activity, and this tells us the activity at the level of the auditory nerve. And we have developed a framework that we can transform this simulated activity in the auditory nerve to the electrical potential that we measure in the scalp, which is what we measure in patients. So we go from the auditory nerve activity through a convolution with a unitary response to whatever we measure in the scalp. And this framework works well, but these models of the auditory nerve are based on physiological data from animals, and then they have been humanized somehow with a number of assumptions. And this works generally well, but not perfectly. And now we are working on improving that model in order to get better predictions. So for example, here the black traces are something called the auditory brainstem response, and the red ones is the original simulations, which more or less follow, but there is some problems with the latencies, for example, of those peaks. And now we are changing things in the model to get much better predictions. And this is important because we want to have this tool and then present a number of stimuli to the model so that we can generate a large simulated data set. And the good thing is that this model, we have all these blocks that represent the function of different cells, and we can impair that. We can change those functions in order to have an impaired auditory nerve model so that we can simulate also impaired evoked responses. The idea is to simulate many of them with different profiles of pathologies, extract some features, for example, the amplitudes or the latencies or so on, and then train a classification model that will link us the evoked response to the cell damage in the cochlea. And so we can have the different profiles. And then the idea is that a real patient comes, we take some measurement, we put it in the model, and then the model will tell us to which of those profiles this patient is more close to happen. So this is a tool to go from the measurement to the pathology, to the individual, to the damage in the individual cells. The second one is now we're going big. Now we are considering large amounts of data. I mean, from the bioinformatics people, this is not a large data set, but for us that we are used to studies with 20, 30 patients, considering this large data set, this is a lot of data. So the idea here is to look and do an analysis to the data set that we have at this hospital, the Rigshospital Hospital in Denmark. This is data collected from 1995 to 2022. I'm considering adults, so 85,000 people, about 300,000 audiometries. That's the main test in audiological assessment, but we have other tests like speech and noise, something called acoustic reflex, some other tests in all those patients. This is the age distribution, so obviously, we have more older people than young, but we have a decent amount of young people as well. And the idea here is to find clusters of patients, something what is called auditory phenotypes, to find groups of patients. And then again, when a new patient comes, we can do all those measurements and assign it to one of the groups that then later we can link to whatever happens inside the cochlea. So we can know more precisely what happens to the different patients. And I can show you some initial results, and I will show PCA results and UMAP results, and that's why I was asking before. So if we do a PCA on all these 300,000 audiometries, we get something like this. The two main dimensions explain that. And this is if I project or if I show the age of the patient, you can see younger people are here, older people go more to the right. And now what we can do is to project known shapes of audiograms. So if I just consider audiograms that are in the good hearing and I go just reducing the hearing, so producing more hearing loss, and I put it in this PCA representation, they just go following the first component, which we know in a PCA is quite related to the mean of the data, right? The second component, what it's showing is the slope of the audiogram. If you have high frequency hearing loss or low frequency hearing loss, you're moving on the second dimensions. So this is a projection of this one, but if I add some audiograms here that have high frequency hearing loss, they go to the north of the PCA representation. And then I can go person by person and assign it to one of these shapes, which are a standard audiogram from literature. So even though we cover a continuous space, probably we can also find clusters within this space. But since our data is a bit more complex than the audiogram, we were exploring nonlinear models as well. And this is the results of the UMAP representation with this same data and this same standard audiograms being represented, again, from good hearing to worse hearing. And what we can do now, or what we think we can do, is since we have patients that have been coming to the clinics over the years, we can trace trajectories. So find a patient and see where in the map he is at the beginning and which position has been transited over the years. So this is the idea. So maybe we find a main trajectory here and another one here and another one here. And this we think can be quite useful in other, and then since we have it over time, we can also probably assign times to this evolution of the impairment. And we think this can be a good tool to counsel patients to say, look, now you're here, but we know that in 10 years you're going to be down here, so do something. Or it can also be a tool to evaluate intervention. So if we know this is the natural trajectory of the impairment, now if we do some intervention like a hearing aid, maybe this produces a change in the trajectory. So this can be a useful tool in the clinics. That's it. Thanks to the collaborators and the funding sources and all of you for listening. Thank you.
Okay, so our next and last speaker is going to be Joanna Lubratska, and also from the same group. And she's going to talk about Chat8, evaluating interactive communication benefits of hearing aids using conversational agents. Just while this is being prepared, please, when you go out, be sure that you sign, if you didn't do it before, you sign the sheet, because then we will use that to generate the certificates of attendance. Okay.
Okay, hello everyone, and welcome to the presentation. Let me first introduce myself. My name is Joanna Lubratska, and I am a senior researcher in the audio technology team at Eurecat. It's a technological center here in Catalonia. We are based in Barcelona. And my background is in a mix of sound engineering, also hearing technologies, and also studies on human auditory perception. And in my current work, it's also a little bit of a mix, but I'm interested in spatial audio, so how we hear the sounds in space. Also virtual reality. This is also a really fascinating tool for me. Also machine learning and for audio processing specifically or especially speech enhancement. And perceptual studies are still close to my heart, let's say. And my connection to Vic is that I have recently started a teaching position here at Vic, and I'm teaching sound engineering and next year psychoacoustics. So yeah, really happy to be here. Today I'm gonna explain you a project, actually an idea of a project that definitely is an example of using AI technologies to solve problems related to audiology. And there's already a lot of AI, a lot of deep learning embedded in those devices, in audiology and in hearing aids. For example, there's already speech enhancement models running inside a hearing aid, for example, denoising or separation of speakers, things like that. Also, machine learning is used to detect the set of parameters that is most optimal for a specific hearing loss or a specific user. So all this is happening, but now today I want to focus on something else. So how AI can help us in the evaluation of hearing aids. So more on the side of actually evaluating the perception while wearing a hearing aid. Okay, and maybe you know this, but hearing, or maybe not, but hearing loss is a very common problem, very common health issue. And this is especially the case in aging societies like Spain. And unfortunately, there's no cure for hearing loss. So once our auditory system is damaged, there's no magic pill that we can take to restore the function of hearing system. But what we can do is we can use hearing aids, and there are kind of assistive rehabilitation devices. And what they basically mostly do is that they make the sounds louder. And as easy as it sounds, the hearing aids have developed very much over the years. So, for example, you can see here this oldest hearing aids, they were basically actually making everything louder. And this must have been really annoying for the people. And nowadays, hearing aids are still making things louder. That's their main function, but they are doing it in a very much more optimal, selective, and intelligent way. Unfortunately, hearing aids still land in the drawer. So people buy them, try them on, and then they don't want to wear them. And now, question to the audience: why do you think that is? Like, why do you think people don't want to use hearing aids? Any ideas? Well, I can tell you one thing is too difficult. There are, of course, many reasons, but one of the most common complaints is that the hearing aid doesn't help me in social situations. So in situations where I actually really need it, I don't need it to watch the TV. I need it when I'm talking to my children and in social situations. And one reason for this, or like, let's say a big picture reason for this, is that the hearing aids are developed and tested in laboratory conditions. And for many years, both the hearing and hearing aids was assessed with passive listening tests. So, for example, if you hear a tone, please press the button, or please repeat the sentence that you have heard. And these tests are good. They give us some insights, but they tell us how well we hear, and they don't necessarily tell us how well we can communicate with others, right? They just assess the hearing part. And so we don't really know how much a person will benefit from a hearing aid in real life when doing those tests. And looking at the user satisfaction, this is really not ideal at the moment. So what we need is some kind of test that will show us how well the person communicates with others. So yeah, how to assess communication. Again, any ideas in the audience? How do you assess if someone is communicating well or not? Maybe some kind of a test, like here, like a language test, right? Someone speaking with the person to check if this person can respond, answer the questions. Well, there are several problems. First of all, we need some assistance. So for each type of test, we need at least one experimenter or a person that will be communicating with our participant. Second, there is a reproducibility problem. So in human conversations, every conversation is different. So now imagine making a study with 100 people, and there's no way of having 100 conversations that are the same, right? And another thing is the scalability problem. So what if we want to control some aspects of the acoustic scene? For example, we want to increase noise or change the position of sounds in space. This would be also really difficult if we had to hire a person to constantly have a conversation with participants. So now we had the chance to think a little bit about this idea and how to test communication. And we thought that actually current technologies allow for creating a virtual conversational agent, so like an artificial human that could be used to test communication. And also there are tools to do it in a controlled acoustic scenario. So now let's go over this graph. So we have our participant, and here we have our virtual agent. And so the participant can speak to the agent, and the voice of the participant is recorded, and then it goes through the speech recognition system, which are now amazing and work in real time. And those speech recognition systems give us text at the output. So once we have text, we can send that text to what everyone nowadays knows and is using, which is large language models like ChatGPT or Llama from Meta, which is a type of tool that can generate natural conversations with humans, right? So we give it text, it gives us back the text. And now the responses from the ChatGPT, we can take it and we can use another deep learning tool, which is speech synthesis. And these models are also becoming very, very good. Like the problem is basically solved almost. So with text, we can generate natural sounding speech. And we can use that speech to feed a virtual agent. And the agents are, these are all artificial agents. They look as well quite realistic, although this is not the most important. The visual part is not the most important here. But we can feed the virtual agent with the speech from the speech synthesis model. And at the end, this agent can be placed in a virtual scene, virtual acoustic scene. So we can create a scene that represents, I don't know, a street like here, or restaurant, or whichever. Sky is the limit. We can imagine what we want. And there are tools to actually simulate acoustics. Instead of just going to a place and recording the thing itself, we can simulate it in a computer. Yeah, so these are using virtual audio techniques. So some things would be solved with such as this system. First, there's no need for assistance. Second, so because the agent is doing the job of the experimenter. Next, we have the reproducibility, because the conversation can be programmed or adjusted so that so it becomes much more predictable than a conversation with real person. It's still a artificial agent. And then the problem of scalability would also be largely solved, because with virtual audio techniques, we can control all parameters of the audio scene. So where are the sources, how loud they are, what is the noise level, all of these things we can control. Okay, and now having this system how would we test communication? That's the most important question, right? So for one idea is that we could create simple communication scenarios, like for example, 5-minute or 10-minute scene in a street. Then another scene would be a restaurant, another would be a museum. So like small different scenarios in which we would give a task to the participant to collect specific information from the agent. And then while doing that or during such experiment, we could measure the communication performance, but how well the person can get the information. So how precise is the collected information? Then we can measure the time that the person needs to get that information, or also the reaction time that the person needs to respond. And also one measure that has already been used in literature, which is the request for a repetition. So how many times the person say, sorry, I haven't heard, can you repeat? So this is, these are some possible metrics. Additionally, in a laboratory setting, we could use some biosignals like a pulse rate or pupillometry in the first phase of this project to test, to measure additional variables that could be analyzed here. And I think, so to summarize, I presented you a basic idea that the idea I think probably is relatively clear, but of course there's a lot to be implemented in the future. And that's why I kind of like this project, because it really requires both it's really multidisciplinary. So it really requires both the technical part, where someone who will implement all the system, but also we need the psychological perspective very much, because we need to design a methodology that will be that will make sense for communication, for testing communication. And then the impact here would be that if we have a method that can quickly assess communication performance, we can use it to guide the settings or guide the development of hearing aids, of algorithms for hearing aids. So those algorithms of, let's say, speech enhancement or hearing correction, they could be tested with these type of tests instead of simple or simpler passive tests. And so eventually, in the long run, the goal is that the hearing aids are more suited for situations where we are interacting with others. And this is what, yeah, I guess every hearing aid user really needs. And with that, thank you very much for your attention. And if you have any questions or comments, yeah, I would be glad to answer.
Well, thank you very much for your presentations. I think that the objective of seeing many different tasks and many different topics has been accomplished. So there is some question by Beth.
So thank you for your talk. It's the one I understood a bit. But what about the position where the sounds come from? Because I always tend to think that this problem with social communication is that the sound comes from different places, not only overlaps, and there's a lot of noise. How would you add this parameter here?
Thank you very much for that question. So, I think, so first of all, it is possible to add a different spatial location. And it is true that often our speaker or the position of our ear against the speaker is not exactly like straight towards the face of a person. So it's good to add this variation of where the speaker is coming from. But I think the first trial would be to create situations that are most natural. So let's say like a conversation at the table where people are seated, let's say around the table. So this represents it represents a situation that can be easily found in real life, and then but it's not exactly like looking at the speaker straight away. So I think this would be like a first trial to just mimic what we see in real life. But of course, we could look at the result as a function of the position of the speaker. But that's, I think, it's a little bit different question. And this research has already been done, but not in virtual reality, not in all the things. But yeah. Well, so well, there are techniques to spatialize audio sources, and yeah, and you can create with the headphones. So when we have headphones, we always have left ear and right ear, and by manipulating the signal in left ear and right ear, specifically by shifting it in time a little bit or adding a little bit of volume to one of the ears, we can create an impression that the sound is coming from a certain direction. So this is how it's done technically. And then you believe that the sound is coming from somewhere, but this is just artificially created effect on the sound. And then there are methods, more detailed methods to do that, so that this impression is really that you cannot tell that it's artificial. This is, yeah, quite impressive.
Some other question? Yep.
Thanks for the presentation. I was wondering about the course you have in Danish hospital. So I was wondering about maybe something has changed due to the emerge of the earphones and everything that might be create another pattern. So I don't know, like if there could be some ideas on how we young people or how people of this generation could improve our hearing or at least maintain the hearing. I think it's just something we won't realize until we lose this. So yeah, it's just some kind of naive thoughts, first about the pattern, and second about if you could have some suggestions about what is the current stage of the hearing health of this generation. Thank you.
But no, keep the microphone because I'm going to ask you. Are you referring to a problem that maybe the profile of patients over the years have changed? Are you referring to that?
Yeah, because I noticed that you project, you embed the data in PCA and also UMAP, and it seems not like I didn't see the pattern like of the current.
So we're considering the whole data set. So we have 30 or 40 years of data, and we are doing an analysis on everything. And of course, there could be some patterns in the data that could evolve over time. I think we have an enough amount of data to later on do a more segmented time analysis. But looking at the literature, when you look at all papers from, I mean, psychoacoustical studies have been done for many, many years, you don't see big changes in how hearing impairment affects to people in different age groups. So I'm I will be surprised if I see a very big effect over the years in different patients. I think it's quite stable. And the second one you said about
no, that's just based on the hypothesis that the pattern might have changed and then the consequences that we are facing. But yeah, thank you.
Yeah.
For the answer.
Morning. It's not a specific question, so sorry, but it's I was thinking all this morning about all the things, about the speak from Thomas, Roger, Laura. But I'm gonna don't say all the names because maybe I forbidden someone, and so sorry. First, congrats everybody. But I always, when I listen researchers, I was thinking how to apply this to the street, how to keep the science to the road. My job is catch science and put this science to the dairy farms, okay? And I think the last presentation, it was, you said, I'm gonna make, I'm so sorry for the name, an hypothesis to do, to apply the science or the investigation to one problem, okay? And that it works today because in dairy cows, so sorry, but I work with dairy cows or with pigs. All the things you speak this morning are applying in the farms, but the people don't know to work with this data. And with simple statistics or with basically programs, Excel or RStudio, we can do little analysis, but our problem is not the number of data because I can store a lot of numbers from cows. Okay, the cows have a big library from data, just 20 years. I can access to this data and I can analyze with simple regression, lot of patterns. Okay, but I think when the doctors or medicine researchers are analyzing data, you stay in a cage, okay? Because it's too are moral aspects, are a lot of things that you can make a problem from another human, okay? In animal dairy science, this is stolen about big companies, and big companies decide what happened. I explained me, so sorry, I'm not gonna go longer, but I wanna set your works impact more longer than you think, because in other parts of the science, we stole all of that and we apply in a cow. But this is make a profitable animal with more profits, animal more efficiency, and we more ecological products from meat and from be alive, okay? And so sorry, but I think this is interesting because when you see human research, if you are a vet, I was a clinical vet, I work in the in the farms, in the computers, and says me, put enter, with a easy thing. I'm gonna set you that things it's applying now in the road. And said congrats everybody. So sorry for the speech or no, the question. You are not scared about the number from your samples because always I see a research, the sample it's too low. I take decisions about 60,000 cows data from a lot of days, milk data about every day from a cow. I can gonna know when a cow gonna be ill because the milk gonna go down, okay? But every day I said to the farmer, this cow gonna go to the slaughter because I know this cow don't gonna produce milk. I was scared with a lot of data, okay? In human research, I was amazing with the brain, with your data or the system prepare more data because you don't have more data. But if I was the researcher, I'm gonna say I was scared because maybe there are a bias, no? So sorry.
Let me just make a short comment first on what you said. Malo, Len, this is a very long discussion and conversation, but what I would say is that I would summarize it in the fact that when you are in the food production industry, everything is about benefits, or most. When you are in the health business, everything is about costs, which means that the benefits means that if I want to get profit off that, don't worry, I'm going to invest whatever because I want to take money from that. But in medicine, it's not the same. So just as an anecdote, last year we presented a project to an open call by the Generalitat on genomics, right? And actually the question was, for many of us here, extremely simple, because they just wanted to have a database, a shared database between the hospitals, okay, on genomics, not on genomics, no, but on particular genes related to some particular cancer types. While all these guys here are going, 10 steps further, okay? So that's the difference. That's a big difference. So in science, in health, it's really amazing because, well, everything, it's the sky is your limit. You can explore everything. But in food industry, it's totally the opposite, no? In general, no? So, whatever. I don't want you to do, a single cell of the cows. I just want you see the point. So I just want to see if that particular antibiotic is going to be cheaper than the other one, and how much do I have to feed the animals with that. So I think that it's a driving force for research in human health, and instead in food production, for example, things are radically different. But still, the data is data, and the tools are very, very similar. So we saw many things, and most of them are, practically the same. So we can talk on tools very easily. So just that the goal is different, no? So, sorry for the speech.
Yeah, I just wanted to mention that there are other research topics in our groups that, for instance, we have a line of research in metagenomics and microbiome analysis that I think that has a strong potential in your area. So we will be happy to talk for possible collaborations.
So yeah, I will reply to your specific question. When Roger asked about the imbalance data sets, the first thing that I said was in the neuroscience field, we took a lot of care in balancing the data just to start with. We would never create new data from there, because that's why we have experimental subjects, and we respect that. Otherwise, we are creating fake data. But as a conceptual answer, we were talking about the augmentation. So if you are talking, for instance, about signal rows recognition, when you augment the data, basically you are just changing the geometry. If you have very few stop signals, you can increase a little bit the scale, squeeze a little bit the data, and that doesn't have a conceptual impact on your analysis. So absolutely no creating data in neuroscience here.
Okay, just to finish, remember that the physicists consider the cows to be spherical. Okay, so that's something important too. Okay, thank you very much, everybody. I think that this has been very good. We're more or less on time, so I promise 1 p.m. and it's more or less 1 p.m., 1:05. Thank you for coming. I hope that you take something home from this, and I hope that you take some context to do. Okay, so people has talked about different things, and now you know that everybody does very, very interesting stuff here. So let's open our minds and get collaborations. Okay, thank you very much.